these hyphae penetrate the cell walls of these plant roots, and then the hypha branches extensively, forming these tree-like patterns that you can see here. Again, the fungus is the dark brown, and that's the basis for the name arbuscular. Arbuscular means tree-like, and so it's that tree-like branching pattern where the hyphae are getting a large amount of surface area of the hypha, or of this branching hypha in contact with the cell membrane of the plant root cell. So these are in direct contact with the cell membranes of plant roots, unlike the case of the actomycorrhizal fungi, which don't penetrate the cell walls and therefore don't get in contact with the cell membranes. So this allows for a pretty free transport back and forth across this fungal plant root boundary, the cell membrane of the plant. And these are some of the earliest fungi to appear in the fossil record. So if you go back to the Silurian, about 460 million years ago, we see the first plant or fungal fossils. They're these things. And it's pretty clear that these associations preceded the evolution of the vascular plants, the ones that have roots and branching bodies above ground, the things that we normally think of as the vast majority of plants. And it's questionable whether the land plants would have ever evolved large branching forms, all the herbs and trees, shrubs that we depend on for life, if it hadn't been for this association with the arbuscular mycorrhizal fungi. So this is, if you take away this association, a lot of plants won't survive. So it's a very important association. It needs to establish very early in the life of the plant when it's a seedling. So this is there are some plant groups that have lost this association secondarily, but it appears to have been a primary association in the evolution of plants. And on the right, you can see that those darker staining areas are those arbuscules, those extensively branching fungal hyphae in association with the cell membrane of the plant root cell. And the cartoon on the right there, that I showed earlier, is trying to make that same point. You can see the dense branching of the hypha there on the cell membrane. You can see it's penetrated the cell wall of the root cell. All right, so now we're going to get to some of the diversity of the fungi. And as I mentioned, there are about 100,000 described species of fungi. And we know that there are 5 major clades, monophyletic groups of fungi, that break out into these 5 divisions or phyla, shown on the right here, with some examples. And there's another group called the microsporidians that we're not going to talk about, but if you're into human health, there are some disease-causing organisms that were actually thought to be some of the earliest eukaryotes that were thought not to have any mitochondria. But it turns out there are mitochondria present. They're just very difficult to see. And we're not going to talk about those organisms, but I do want you to know that it turns out that they are fungi, and they might represent a 6th major group here, but it's still a bit uncertain. We're going to focus on the 5 shown here. And the earliest branching lineage that you see up at the top are the so-called chytrids, members of the Chytridium mycota. And this represents only about 1,000 species of the 100,000 that are described. So only about 1% of the fungi, but they're really important, both evolutionarily and ecologically. Evolutionarily, they're important because they retain some of the features that are shared between fungi and animals. And so they allow us to see that connection with the animals and some of these other members of the larger clade that they belong to. And one of those characteristics is the presence of flagellae. So they actually have a flagellum, a flagellae, that are posteriorly located on the unicellular gametes and the zoospores. So the reproductive cells, the spores and the gametes, have a flagellum or a flagellae attached at the back end, the posterior end, just as in animal sperm and in the close relatives, unicellular relatives of fungi and animals. And I mentioned last time that this is one of the diagnostic traits for that clade that includes the animals and the fungi. This is the only major group of fungi that shows that trait of having those flagellae. The other major groups we're going to look at, they don't have any flagellated stages. There are no flagellae present. So chytrids mostly occur in aquatic situations or in soil. And many of them are unicellular, but some of them do have branching filaments. And they share these other features with fungi with regard to those hyphae. So I'm going to turn now to you can see a sporangium releasing some of these zoospores. See for yourself that these things have motile spores. So here, out of the tip, you can see release of, there's some zoospore. There's a zoospore swimming around there. There's another one. So they can disperse, the reproductive spores can disperse by swimming around, which is very unusual for fungi. Another important aspect of chytrids is some of them are in association with animal guts, with some ruminants that they can help with the digestion of otherwise difficult to digest substances. So chytrids can have mutualistic associations. Unfortunately, they can also have parasitic associations. And one of the most tragic developments of the last quarter century or more, I guess now it's more like, well, yeah, about a quarter century ago, is this disease that's been spreading around the world called chytridiomycosis. It's a chytrid disease that affects amphibians, and it's led to a wholesale mass extinction of the amphibians worldwide, especially the frogs. It infects their skin, and it eventually kills them. There are some frogs with some resistance, but there's already been a large number of complete extinctions of really distinctive groups of frogs, species of frogs, and it's spread throughout the world pretty quickly. There are a number of causes of amphibian decline worldwide, but this is one of the most significant and one of the most pernicious. No one's figured out a way to stop this chytrid, although it's an active area of study by members of the Museum of Vertebrate Zoology here on campus, in fact, in this building. In the Sierra Nevada lakes, where predatory introduced trout into the high Sierran lakes was causing a lot of death of our yellow-legged frogs, our native frogs there. Just shortly after they eliminated a lot of the fish from those lakes, the chytrids arrived. And it's not uncommon to find dead frogs floating in alpine lakes in the Sierra, which was never seen before. And they're completely extinct now from much of the Sierra Nevada. So this is happening worldwide, and it's a really major problem. It's thought to have possibly been introduced by African clawed frogs, which were historically used for pregnancy tests. If you inject the urine of a pregnant woman into an African clawed frog, a female clawed frog, it will lay eggs within 8 to 12 hours. And it's a pretty reliable pregnancy test. They're not used for that purpose much anymore, but these frogs are widely distributed. They're also used in labs as experimental organisms. That's possibly a vector of how this has gotten transported around and then carried around by people and other. Anyways, it's a real problem, and it's caused by chytrids. All right, so the next major group of fungi, that was the Chytridiomycota. Now we're going to look at the zygomycota, which is the next group branching off. And the zygomycetes, as they're called, are things you're probably more familiar with in chytrids. They have about the same diversity. There's only about 1,000 species, very low diversity compared to some of these other higher fungal groups. But they're pretty ubiquitous, and they're real they're really adept at dispersing and invading fresh produce, like the kinds of things that you buy at the store and put in your refrigerator. And they're great at infecting bread too. And so some of the common bread molds, common black bread mold in particular, you see here in the lower left, you can see the mold, the furry covering that you're probably familiar with if you left bread around for too long. These things grow fast and they quickly invade their substrate and grow even on the outside, so they're called molds. In this case, these are capable of forming, well, I should say that fungi do have sex, but they don't have male and female dichotomy of two different types of sexual partners. Fungi have mating types, and with any given species, there can be a large number of mating types that are compatible. Members of the same mating type, though, are not compatible with each other. But fungi do have large numbers of these different types of sexual partners that can pair up in different combinations. So it's pretty different than animals and even plants. But this is a group in which we have the production of pheromones, like in other fungi, that causes the reproductive filaments, what we call the gametangia, to grow toward each other. And you can see here on the right, those two clear enlarged bulbs on either side of that black object are the gametangia that have come together. They've had fusion of their cytoplasm, and then we've had the formation of this big dark knobby zygosporangium, sometimes called a zygospore, within which we have the fusion of nuclei and subsequently meiosis to produce sexual spores. So the significance of this is sexual reproduction in these organisms. They don't produce a large macroscopic fruiting body like we see in the higher fungi, but they do have this very morphologically distinctive structure that's resistant to desiccation, to drying out, to environmental extremes of various kinds. And so it can be a resting stage where these, shortly after we have the beginning of sexual reproduction occurring here, this thing can basically wait out periods of bad environmental conditions, but eventually, when conditions are right, it will split open and produce a sporangium that will then disperse these sexual spores. And sexual reproduction is not nearly as common in these organisms as asexual reproduction. Asexual reproduction is spectacular in these things. In some of them, for example, like the pilobulus shown here. So they can produce huge numbers of these little sporangiophores, these sporangial stalks with a sporangium on the tip. This is maybe about 1 centimeter long maximally. But they can shoot these sporangia, these dark blue objects here on the tips, they can shoot these sporangia about 2 meters. That's an incredible distance for something that has a sporangium that's only about a tenth of a millimeter in length. That's an extremely great distance, comparable to best artillery. And if you're to scale this up to human size, it'd be like shooting yourself out of a cannon at 100 times the speed of sound. So, I mean, that's not a completely fair comparison because gravity doesn't affect them as much as it would a larger object. But nonetheless, these things can really get their spores dispersed widely. In this case, these things grow on dung of animals, and they can shoot those sporangia to a blade of grass, for example, where they'll glom on, and then a ruminant will come along, eat the grass, and then when it passes through the gut, the fungus will have its ready-made food source right there with it when it lands on the ground and can grow up through that wonderful substrate. So that's the zygomycetes. All right, so the third and really important group here, but least diverse in terms of numbers of recognized species, is the glomeromycota. And this is a group that used to be considered part of the zygomycetes. Part of the difficulty in classifying these things was that there's no sex that's ever been seen in them. And the sexual structures are the ones that are most diagnostic for deep relationships in fungi. But no one's ever observed sex in these things. But nonetheless, molecular data have shown that they're more closely related to the higher fungi we're going to look at in a moment than they are to the ones we just showed. And these are the organisms that are responsible for these arbuscular mycorrhizal associations. These are the arbuscular mycorrhizal fungi. So even though there's only a small number of species known, well under 1,000, these are crucial ecologically for the fitness of plants worldwide. So a really important group, and we've already talked about their ecology a bit. All right, so now we're going to get to the so-called higher fungi, and these are the ones that produce some of these really striking fruiting bodies. There are two major groups, and this first one are the ascomycetes, the ascomycota. And the reason they're called ascomycetes is because they produce these sacs. You can see in the center image there, you see there are about 8 spores that are surrounded by a translucent membrane. And that entire structure is called an ascus, the so-called sacs of the sac fungi. This is a group called the sac fungi, the ascomycota. So the production of these sexual spores within these transparent sacs are what gives this group its name, and they're a very distinctive characteristic of this group. We're not going to get into the reproduction of these in detail, although it's in your reading, and I encourage you to look at it in the book. But I do want to mention one thing that is important is that these so-called asci, which is the plural for ascus, are found oftentimes within these fruiting bodies called ascocarps. Okay, and these ascocarps have a wide diversity of forms, including things like morels on the left, choice edibles, and you can see the morels on the left and the cup fungus on the right. They share that characteristic of having these depressions in their surface. The cup fungus is called a cup because the center of it is hollow, and you can see the morel has these depressions all over the outer surface. Those surfaces are covered with these asci that produce the spores. Truffles are also part of this group. Truffles have underground ascocarps that produce asci. So there's a lot of different types of these fruiting bodies. Truffles are by far the most valuable. The white truffle on the open market now sells for about $15,000 a pound. So it's quite valuable. There is cultivation of truffles that's ongoing, but some of these are not easy to cultivate. So really just choice edibles and a lot of different forms of these different types of fruiting bodies. So the ascocarps, or I should say the ascomycetes, and the next group that we'll look at next time, have their hyphae in this category on the left. They have this form called septate hyphae. So they do have the hyphae broken up into partitions or compartments. And this is important when we look at their reproduction next time. There are unisexual higher fungi. Both ascomycetes and basidiomycetes include species that we call yeasts, these unicellular taxa, and they reproduce asexually by budding. So you can see some of this budding going on here, for example, where we have mitotic division producing a genetically identical progeny cell that's smaller than the adult. and grows to a larger size. This kind of asexual budding can occur really rapidly. Yeast colonies can grow quite quickly. This is a group that doesn't represent very many species. There are only about 1,000 species known of yeast, but there's probably a lot more diversity that's undescribed because there's not a lot of morphology here to base descriptions on. Basidiomycetes are the next and last major group of fungi. The ascomycetes include about 2/3 of the described species of fungi, about 65,000 species. The basidiomycetes are about half as many, about 30,000. And these are the so-called club fungi that include the true mushrooms, as well as things like puffballs, bracket fungi, jellies, like you see there on the left, on the right, lower right, and things like rusts and smuts, some really important disease-causing organisms. So a wide diversity of forms, especially of the reproductive phases of these organisms. And these basidiocarps, the fruiting bodies, are often formed out at the periphery of the mycelium. So that entire area that's enclosed by those fruiting bodies, the so-called fairy ring, which in medieval times was thought to indicate the circle in which fairies would assemble after dark. That whole area was, at one time at least, covered by mycelium. So at the center, it started somewhere about the center of that ring and grew out in all directions, radiating out. And these fruiting bodies then are formed out at the periphery where the mycelium is at its growing edge and coming in contact with virgin food sources. So it's going to mobilize nutrients most easily in places where it's right in contact with the richest food sources, and that is most beneficial if you're producing these big fruiting bodies. Okay, so we'll get into the basidiomycetes more next time, and you'll be seeing some of these organisms in lab this week. Okay, so see you Wednesday. We're at office hours, hopefully.